Leukemia is a cancer of the blood cells. There are 4 different types of leukemia. It can be either acute or fast-growing, or chronic or slow-growing, and either lymphocytic, affecting lymphocytes, or myeloid, affecting myeloid cells. A lymphocyte develops into a type of white blood cell, while myeloid cells can develop into white or red blood cells as well as platelets. Let's take a closer look at chronic myeloid leukemia, or CML. CML is often caused by the translocation between chromosomes 9 and 22, resulting in what is known as the Philadelphia chromosome. Somebody with CML caused by the Philadelphia chromosome is said to have Ph-positive CML. The BCR-ABL fusion protein that is formed from this translocation is constitutively active, meaning that it's always on. The BCR-ABL protein is a tyrosine kinase, meaning that it is an enzyme that phosphorylates other proteins at a tyrosine residue. Adding a phosphate to certain proteins activates signaling pathways that are involved in cell growth and prevent apoptosis. Since this enzyme is always active, this results in the overproduction of Ph-positive blood cells that build up in the bone marrow and blood. This makes it very difficult for normal function of blood. To combat Ph-positive CML, a common drug used is Sprycel, which is manufactured by Bristol Myers Squibb. The active ingredient in Sprycel is Dasatinib, designed to inhibit the function of BCR-ABL kinase. The BCR-ABL protein works by transferring a phosphate group from an adenosine triphosphate molecule, or ATP, to a tyrosine residue on the receiving protein, resulting in an adenosine diphosphate molecule, or ADP, and an activated protein. Dasatinib works by displacing ATP in the binding site of BCR-ABL kinase. Since it binds directly to the substrate site, it is called a competitive inhibitor. Since it doesn't have a phosphate group to donate, it doesn't activate the signaling pathways that cause abnormal cells to grow and cancer to flourish. The ATP binding site is a hydrophobic cleft between 2 lobes of the protein. In the BCR-ABL ATP binding site, the sulfur-containing thiazole ring sits in the same location as the adenine group of ATP. The chlorine-containing ring sits in a hydrophobic pocket that is not normally occupied by ATP. The rest of the dasatinib molecule extends toward the pocket opening, with the hydroxyethyl group on the non-aromatic piperazine ring being solvent exposed. Within the active site, dasatinib forms many non-covalent interactions with the wild-type kinase, including a hydrogen bond between the amide nitrogen of methionine 318 and the nitrogen on the amino thiazole ring. A hydrogen bond between the carbonyl oxygen of methionine 318 and the 2-amino nitrogen. A hydrogen bond between the side chain hydroxyl group of threonine 315 and the amide nitrogen. A heteroaromatic interaction between the backbone carbonyl oxygen of glutamate 316 and the C4 carbon of the amino thiazole ring. A heteroaromatic interaction between the backbone carbonyl oxygen of methionine 318 and the C5 carbon of the pyrimidine ring. An off-centered aromatic pi stacking between the pyrimidine and aminothiazole rings of dasatinib and phenylalanine 317. And lastly, van der Waals interactions such as a hydrophobic cleft created by leucine 248 and glycine 321 where the pyrimidine ring sits, and a hydrophobic pocket composed of threonine 315's methyl group, methionine 290, valine 299, isoleucine 313, and alanine 380, among others, where the chlorine-containing ring sits. Due to dasatinib's affinity to the ATP binding site, it has been an effective anti-cancer drug in patients with pH-positive CML. The death rates for people with CML have been declining over the years, and it is no doubt due to drugs like dasatinib as well as many other treatment options available. Through continued drug research and development, this trend should continue for many years to come. Thanks for watching.